It will be vocab heavy, just heads up.
We'll just go have everything with the test.
Of course.
Please don't make it that Friday of Easter weekend.
There's no class that day.
Oh, yes, seriously?
Yeah, we have Good Friday off.
I'm going to the beach.
No, I already went there, it was too
Will
cold.
it be COVID?
All right.
Said there's no school Friday.
There's no school on April
Next Friday.
2nd, next Friday. Good Friday is so news. Good Friday is next Friday. No, we do not get Monday. No, you'll be here on Monday. So, and your exam will be the Monday after that. We've got 2 more weeks, I believe. Yes. Leftover fish tacos. That's a hard task. No, that's just no.
He'll bring you a whole
Spanish man.
No. All right, just come to class. All right, chapter 12. Patterns of inheritance are a result of the behavior of your chromosomes in meiosis. We're just going to keep hearing that a bunch for like the next 2 chapters. Okay, starting with heredity. We inherit stuff from our parents. That's easy, right? Now, we're going to talk a bit about kind of the historical ideas about heredity because we're a bit weird, right? And then we're going to talk about Mendel. You may or may not have done like the Mendel stuff in high school. So we're going to go through pea plant experiments, Punnett squares, all that fun stuff. All right. So pre-20th century understanding of heredity, right? We don't know what DNA is. So we know that heredity occurs within species, sure. Baby rabbits look like adult rabbits, fair. And traits are transmitted from parents to offspring. I look more like my parents than like somebody else's parents, sure. But they, not having a good idea of DNA or genes or those sorts of things, kind of filled in the details in a bit of a fuzzy sort of way. So heredity occurs within species. And so you have blending of traits within fixed, unchanging species, right? Because in their heads, species didn't evolve or change over time. So you just have this blending of traits, right? And then also that these traits were transmitted again from parents to offspring, but since they didn't have any kind of concept of genes or DNA, they thought it was through fluids, usually blood, right? So this is where the term bloodlines comes from, this idea that like the information for kind of what you look like and what you are is literally in your blood, right? Which is not correct, but it's what they thought. But if you think about this for a minute, it doesn't make sense because if no variation is entering from outside a species and all you're doing is blending traits between people every time, right? Eventually you would just see no variation, right? You're just kind of blending and blending and blending. Like if you were blending a bunch of paints, you would just eventually end up everything's brown, right? So something else has to be happening, but they didn't think that. All right. So most of the experimental work that kind of picks apart all of these patterns and models of inheritance have to do with hybridization. Experiments in hybridization is just crossing either or interbreeding or crossing 2 different species or 2 genetically different individuals of the same species. Crossing individuals of 2 different species is complicated and generally just does not work. We're going to focus on variable individuals of the same species, so more pertinent to our purposes. All right, so words that we will use. Parent strains, right? The parents, the things that we're breeding together give rise to offspring or progeny. I tend to say progeny just out of habit, but offspring works. And these strains frequently for scientific purposes are true breeding, what we call true breeding strains. And a true breeding strain is just, right, kind of like a pedigree, like a line of individuals. We'll be talking a lot about plants, right? So if I have a tulip, I have a red tulip, and no matter how many times, right, I kind of propagate this red tulip, I only ever get red tulips, right? It is true breeding, right? When they are bred kind of with themselves, they remain uniform. Lab rats are another example, right? Lab rats are kind of bred into strains so that, right, if I purchase some particular strain of lab rat, it will only, like if I breed them together, they will only reproduce individuals basically identical to themselves. True breeding. It's similar to purebred. Yeah, it's kind of like the extreme form of like a purebred dog or something, right? Because you still, like in like dog breeding, you still have some amount of variation left in there. There's other problems when you become true breeding, but yeah, it's a similar idea. And so you can take these individuals of these strains and breed them together. Those are called crosses, right? So the interbreeding or mixing of individuals is called crossing, right? So if I'm talking about crossing pink flowers and white flowers, right? I'm going to breed these pink flowers with these white flowers and see what happens. I cross them. I cross pink with white. So we tend to just put a little X. You'll see that when we start kind of writing this stuff out. All right, so a pink flower strain crossed with a white flower strain. It means I'm breeding the two together. Okie dokie. So, like I said, a lot of this is done in plants just because plants are easy. They don't move. You can kind of manipulate them a lot. So some experiments in the 1700s crossed two different strains of two different kind of breeds or lines of tobacco. And so, right, we had tobacco A looked like this, tobacco B looked like this. When we crossed those two strains, the offspring, the progeny of that cross didn't look like either of the parent tobacco plants, which is not what you'd expect, right? Given kind of our old idea of heredity, that we should just kind of be blending their traits and get some kind of mix in the progeny, right? But when in this experiment, so we have purple tobacco and white tobacco, I'm just gonna make up colors, right? We bred them together and we got blue tobacco. It's not a thing, right? When they bred those offspring together, some of them looked like those cross ones, the blue tobacco, but there were also purple and white tobacco. I'm just making a bad at this point, right? So there are other things going on that are causing offspring to be more variable than you would expect if we're just kind of like mixing up the traits of the parents and getting some kind of intermediate form, right? So pea plants. I'm going to spend a lot of time talking about pea plants. In the 1800s, T.A. Knight crossed two true breeding, right, strains of green pea plants, of pea plants. One was a yellow pea plant and a green pea plant. Okay. So the yellow pea plants, if they were just kind of bred together on their own, they only produced yellow seeds. The other one only produced green seeds. When they were bred together, all of the offspring only made yellow seeds. Green seeds gone completely, even though one of the parents definitely had green seeds. All right. But if you took these yellow seeded offspring, what is that? Getting the wrong arrows. All right. These yellow seeded offspring and cross them with each other, right? Cross one hybrid with the other hybrid. You got mostly yellow seeds, but now you got some with green seeds. So it's like the green seed trait was kind of like hiding up in these other guys, right? Now, having this where some of your offspring are one way and some are another is referred to as segregation, right? We didn't see like light green seeded plants or green and yellow speckled plants, right? We cross these plants and we have yellow seed individuals and green seed individuals. The trait segregates among your offspring. Yeah. Okay. It will become important. Yeah. So Mendel. Gregor Mendel was an Austrian abbot. So he lived at an abbey. Traditionally, abbeys have these very large, lovely gardens. And so he spent a lot of time breeding pea plants. Lots and lots of pea plants. And the thing he did that none of these other guys were doing, which seems kind of obvious to us, but it just wasn't kind of a habit of scientists at the time, was that he quantified his results. He actually like counted what he was getting. Like, I'm getting 10 yellow pea plants and 3 green pea plants and that kind of stuff. He actually like recorded his data. Actually novel, kind of groundbreaking at the time. So why peas? Because it was easy, basically. So the benefits of working with peas, there was a lot of people who had already been working on peas, right, as we mentioned. There are a lot of pea varieties, 34 different kinds of pea plants he had access to. They're small, they're easy to grow, they grow fast, and you have male and female parts in the same flower, which allows you to do kind of a lot of manipulations. It's easy to let them self-pollinate. You can kind of remove parts from one to make sure that it doesn't self-pollinate, make sure it breeds with some other plant, right? You can do kind of weird things with plants that you can't do with other organisms, right? So they're just kind of a handy thing to work on. So his experimental design using aforementioned pea plants. So a couple of things he did that were smart choices is he only focused on a few traits, right? He didn't like look at every single aspect of a pea plant, right? That would get kind of messy, right? And he made sure that the traits that he chose to look at were kind of in little complementary pairs, right? So he wasn't comparing like yellow seed plants, like was a plant, did it have yellow seeds or was it tall? Right? That doesn't really make sense. So he did like, does it have purple or white flowers? Does it have yellow or green seeds? Right? That kind of thing. Right? So you have these complementary pairs of traits, right? So he created true breeding lines of plants, right? So he bred them over and over and over again until he had plants that only ever made purple flowers or only ever made yellow seeds, etc. Right? He made sure that they were true breeding. And so his main experimental design was to take these different true breeding strains and cross them together, right? Cross a true breeding purple flower plant with a true breeding white flower plant and see what the offspring look like, and then record all his data, right? And he did this with reciprocal crosses, which we'll find out, I believe, in chapter 14, why that becomes important. Right, so basically he just made sure, right, you've got two plants, they both have male and female parts. I'm going to make sure that I do a cross where the male of this plant fertilizes the female of that plant, and then the male of this plant fertilizes the female of that plant. So everything gets kind of mixed every which way, right? So he crossed true breeding strains using reciprocal crosses. Right. And then he allowed those hybrid offspring to self-fertilize for many generations, recorded, blah, 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 blah. Right. Most importantly, he recorded everything. Right. So he recorded the results of every experiment he ever ran, so he could then like look at the results, compare them, do kind of mathematical statistical analyses and that sort of thing, which was this kind of surprisingly very new idea. All right. So again, he was the first to quantify these results. And the mathematical analyses that he did, right, on these pea plant experiments led to the models of inheritance that we still use today, right? So these kind of models of the way that now we know genes and alleles of genes behave still works, right? You can still apply Mendel's inheritance models to these kinds of inheritance studies that people do now. It's kind of cool. All right, monohybrid crosses. So we're going to start small and work our way up. So we're going to talk about monohybrid crosses, what do we get from monohybrid crosses, talk about Mendel's principle of segregation, which is kind of a conclusion that he pulled from these monohybrid crosses. And we're going to compare all of these results to what we know about meiosis and see that there's a relationship between the two. All right. So a monohybrid cross is a cross, right, between two true breeding strains that follows basically a single trait with two options, right? So monohybrid crosses are nice. They're very kind of tidy and simple, right? I'm looking at this single trait. This single trait will either look like A or like B, and that's it. And that's all I'm looking at, right? So pink versus white flowers. So in the pea plants, Mendel basically had a set of 7 traits that worked this way that he would kind of use, right? Whether it was flower color or seed color, seed shape, right? These things where you had a single trait that would show up in 2 different ways, but only 2 ways, right? Easy to observe, easy to record, frequently based on like very obvious shape and color. When you're designing scientific experiments, it's nice to keep it as simple as possible. It makes your life easier, right? We're going to focus on flower color. That's kind of like his kind of baseline, the one that everybody talks about, his kind of model experiment, looking at these purple and white flowers in pea plants, right? So more words we need to do this. When we start doing these kind of series of hybridization experiments, right? We've got these true breeding plants, right? Step 1, I've got purple flower plants and I've got white flower plants. Those are my parent strains, right? The parent plants of the whole experiment. When I breed those 2 together, their offspring, right? The offspring of the parent plants are called the first filial generation. Nobody says first filial, just F1, right? So my F1 generation are the offspring of my first cross. So parent plants, F1 generation. When I cross that F1, like F1 individuals with other F1 individuals, it's F2, logically enough. You could keep going. We're not gonna go past F2. You can go F3, F4, F5. You can just keep crossing these things together ad nauseum. We'll deal with parents F1, F2, right? So children, parents, grandparents, right, is kind of the level we're looking at.
So two is grandkids.
Basically, yeah.
And so that two would be grandkids, right?
Yeah, so you've got like parents, their kids, kids. So yeah, so if you went the other way, you've got like kids, parents, grandparents. We're kind of dealing in a little 3-generation stack. So parents, F1, F2.
So wouldn't it be just F? So you don't even classify the originals.
Parent strains are just the parent strains. It's just whatever you start with, right? Because we know what they look like. Your kind of experimental results are the F1 and the F2.
Okay.
Yep, yeah, absolutely. Now, so he was doing these crosses, recording results for his F1 and F2 generations, and he referred to the traits he was looking at, right? Each of these pairs of traits as dominant and recessive. And what that meant, right? So if I cross, like in our seed color ones, right? We had yellow seed plants and green seed plants, parent strains. In the F1, they all had yellow seeds. And so in Mendel's model, you call that trait yellow dominant. So the trait expressed in the F1 plants is dominant. The trait that you don't see in that experiment was the green seeds, right? We didn't see any green seeds in the F1 in that experiment, is recessive, right? It recedes, it kind of vanishes for a minute, right? And for all 7 of those traits we saw on that last slide, not that you need to memorize all 7 pairs of traits, but for all 7 of those traits that Mendel was using, 1 of them was dominant and 1 of them was recessive. To some degree, we'll see when we get into chapters 13 and 14, he was kind of just lucky, but there are traits that work like this, right? Where one, you have kind of 2 options, one is definitely dominant, one is definitely recessive, right? So, flowers. So you had white flower pea plants, purple flower pea plants. We're going to cross them, right? So we just put that little X in the middle. I'm crossing my 2 parent strains, crossing white flowers with purple flowers. My F1 is all purple, which means the purple trait is dominant, absolutely, right? So all my F1s plants have purple flowers. I'm going to cross F1 plants with other F1 plants to create my F2 plants. And now those are mostly purple, but a few of them are white. None of them are pink or like purple, white, splotchy or anything like that. There's no blending, right? We're basically just kind of ruling out blending at this point, right? So we have, most of them are purple, but now we've got a few white again. That recessive trait kind of comes back around in the F2 generation. So what is going on? Mendel thought correctly that the proportion of purple to white, right, like the actual numbers of purple plants versus white plants would be informative. And it 100% was, right? So when he went back and counted all of his F2 generation individuals, 75% of them are purple, 25% of them are white, always. Same with the green and yellow pea plants. 75% of the F2s would have yellow seeds, 25% would have green seeds, right? For all 7 of those traits, the F2 would show up in the same ratio of 75% of the dominant trait and then 25% of the individuals with the recessive trait. Right, so you've got a quarter of those F2 progeny expressing that recessive trait that was lost, that looked like, right, appeared to be lost in the F1 generation. Same for all 6 traits. All right, so he looked at this F2 generation. All right, we've got F2, 75% were purple, quarter of them are white. If he bred the white ones with white ones, right, those that had the recessive trait, they were true breeding for white, only got white flowers out of white flower plants if you bred them together, right? For the purple ones, right, so let's say for any 4 plants, on average, 3 are purple and 1 is white. White one only makes white plants. It's a true breeding plant. Of these 3 purple, right, the other 75%, one of them would be true breeding purple, would only ever produce purple plants. But these other 2, the other 50% of the progeny, the other 2/3 of the purples, sure, would create a mix, right? So these 2 purple plants would then make a mix of purple and white plants, right? So is that 3-to-1 ratio completely accurate? I mean, it is in the sense that you see 3 purples for every 1 white, but what's actually happening, right? Because they're not behaving the same way, so they must not be truly identical, right? So the actual breakdown, right? Here's my parent strain, breeding purple, true breeding white. Breed them together. F1s are all purple. If I start breeding these F1s together though, 3/4 of them are purple, but 1/4 of them is white. Any individual that's white is true breeding white. End of story. Of these last 3/4 of everybody, right? 1/4 or 1/3 of the purple ones are true breeding purple. The other kind of 2/4 of my progeny are purple, but their offspring are a mix of purple and white. So basically I have 1/4 that are true breeding recessive, 1/4 that are true breeding dominant, and then half of them appear dominant but are not true breeding. So it's a 1:2:1 ratio. All right, so given all of this information, Mendel made some conclusions. One was to get rid of that blending idea, right? Plants do not produce progeny of intermediate appearance, right? We don't get pink flowers. We don't get lime green pea seeds for the traits he was looking at. So no blending. Conclusion one, blending's out. For each pair of traits he looked at, one was not expressed in the F1 generation, but it would reappear in F2 in some of them, right? So this means, and it was not a thing that people were really aware of at the time, you could have a trait present, but not expressed, which we kind of intuitively know, right? You can be carrying genes that you're not exhibiting the traits for, right? There are things you may not know you have that you can pass on to your kids, right? That they might express, that they might actually show, right? We'll come back, I think at the end of chapter 14, we come back around to kind of human examples of all of this stuff. It's just easier to talk about pea plants. All right, so a trait can be present. You can have a gene for something and not express that gene, not actually have the characteristic that you have a gene for. 3, traits are segregated among progeny, which is just kind of one of those kind of duh things, like some of the progeny have one trait, some of the progeny have another trait. It segregates among the progeny. Easy enough, right? Last conclusion, the F2 generation expressed, you saw, right? If you look at them, 3/4 of them show the dominant trait, 1/4 of them shows the recessive trait, right? So that recessive trait comes back a generation later, right? And this 3:1 segregation ratio, right? Segregating 3 purple plants for every 1 white plant is known as the Mendelian ratio for monohybrid crosses, right? And that still works today. For these traits where you have kind of 2 options and one's dominant and one's recessive, the Mendelian ratio applies to them. All right. We will move on to Mendel's model on Friday. So I will leave you there.